Release for short. And the title of my talk was How the Ghosts of Our Climate Past Inform Our Climate Future. And so the pictures that are in my opening slide here, my title slide, these are pictures of living forams. These are photographs that I took. I study living ones that we capture out in the ocean, but they live everywhere in the ocean and they become fossils on the ocean floor. And scientists can obtain samples from the seafloor, and from the composition of their shells, we can reconstruct the temperature of the past. So I also sort of, like to think about this along the lines of how do we take the temperature of ancient oceans? I'm gonna tell you how we do that today. So I'm just gonna get started. Just a quick overview of what I'm gonna talk about today. So I'm gonna start by giving sort of a perspective of modern climate change. What do we know about how the Earth's temperature is changing and what's happening to ice and the CO2 levels on our planet? Modern climate change, in this sense, I'm referring to what I call the instrument record. What can we go out as humans on this planet, go out and measure? And how far back in time do those measurements go? They don't go back far enough. They go back about 150 years. And so because of that, we need a paleoclimate perspective on what the temperature and the CO2 and things on the planet that are changing today. And we get these from climate proxies. And I'm going to talk about what climate proxies are. And then I'm going to give a brief intro to how my research plays a role. I study them in the living ocean. I'm going to talk about how we do this, where we collect them. And then, because this is a talk that, a lot of people who are here today are from Oregon or Corvallis, and some of my research has taken us off the coast of Oregon. And so I'm going to talk about what's been happening off the coast and what we've been doing over the past few years. So the two pictures in this slide off to the corner here, these are forams. These are pictures that I took in the field, and they look pretty big, but forams are actually microscopic. They're about the size of a grain of sand. And so for perspective, I've put a 12-point font, a little dot, a period that you'd have at the end of a sentence. That would be a very, very large foram. To give you a sense of how big they are, they're really quite small. They're about the size of a grain of sand. But yet they're small but mighty is sort of how I like to think of them, because we can get a lot of information about them, or about the planet from them, I should say. So I'm going to start by talking about what we know about how the Earth's climate is changing. And so to get us started, I thought I would show what we know about the temperature on the planet from the instrument record, or thermometers. These are measurements that people have been taking on our planet. This is a figure from NASA, GISS. And essentially what this is showing is the black line in the middle here is the average sort of running mean through about 140 years worth of temperature data. Each individual dot here is the temperature for a single year. We've got the running mean in the center. And we can look at this and say, okay, well, when did we start taking the temperature of the Earth? We began looking at this record about in the late 1800s. Overall, we see a warming trend. We see some wiggles in here. This is sort of decadal variability where we have, a little bit of a decrease in temperature here, an increase here, but the general trend is in a warming direction. So we know even in the last 180 years of us being able to measure the temperature of the planet, that the planet has gotten warmer over the last 150 years or so. 20 of the warmest years since we've been taking, or 19 of the warmest years since we've been taking the temperature of the planet have occurred in the last 20 years, 19 in the last 20 years. The only year warmer is this little dot right here, which is 1998, which happened to be an El Nino year, and it was a very warm for that reason. So we have this warming trend over the last 140 years or so. 2020, it was just announced, it was the second warmest year on record since humans have been taking the temperature of the planet. So we see this and we say, okay, it looks like the temperature of the planet is getting warmer. So what about changes in other portions of the planet? We know that the temperature is changing. So I wanted to talk a little bit about how areas on our planet are responding to this change. And so one of the ways that we can really see what's happening on our planet is looking in the Arctic. So this figure here, it looks like a bunch of squiggly lines. Essentially what this is the average September minimum ice extent in the Arctic. So this is like after the summer ends and as much ice has melted up in the Arctic as possible. We measure this, and we do this with satellite data. The satellite record for measuring ice in the Arctic began in 1979. So we've got about 40 years of data showing us what's been happening in the Arctic with ice extent. Again, overall, we see a change. So we know that temperatures are going up on the planet, and in response, we're seeing things like ice on the planet melt. And the rate of change is about 13% per decade. And some scientists would even suggest that the rate of change is increasing. If you can see my mouse here, there's sort of a change here. We probably don't have enough data to say that that's a robust change, but it has been predicted that we are going to see an ice-free Arctic at some point in time in the near future. And just like 2020 being the second warmest year on record, we also had the second lowest ice extent on record just this past year. So we can look at this in another way. We can look at what's happening in the cryosphere by looking at how thick or thin sea ice is. So this is a satellite measurement showing a measure of how thick the ice is. And on the bottom, it equates thickness to the ice's age. So the white colors, that is old ice. That's ice that's been in the Arctic for, you know, on this graph on the bottom, it says +4, but it's actually about, some of that ice is on the order of 9 years old. And there's a lot of it. So this is March of 1985. And there's a lot of old ice in this region. Oops, wrong way. The dark blue, this is something that is called first year ice. This is ice that formed during the previous winter. And what's significant about first year ice is that during the September minimum events, that ice is typically the first to melt away. And so how are things changing? So this was 6 years after measurements began. And here we are, March of 2018. And we can see that there's a pretty significant difference in the Arctic in this region, or in the Arctic in general, in terms of the ice is becoming thinner. There's been a significant decrease in the amount of old ice and a significant increase in first year ice. And because first year ice is more prone to melt the following year, this makes the Arctic region pretty sensitive to changes in temperature as the planet continues to warm. So this is March of 1985 compared to March of 2018. And up in the corner of the screen, this talk will be shared, so you don't have to write this information down, but I've provided some links because NASA has done a really good job of animating the change that we see. And so you can look these up on Google, or you can wait until this gets posted and take a look at sort of how this has happened through time. And I also wanted to just point out some, we've got like some winning data in 2020. We actually had some record warming in a town in Siberia that had record warming for a week. It was 100.4 degrees at a small town in the Siberian region of Russia, of this area, I'm losing myself here. What we're seeing here is warming significantly in this region, and 100.4 degrees is a significant warm period of time just this past year. So we know that the Arctic is also responding to changes in our climate. And so the last bit of the sort of modern record that I want to talk about is looking at atmospheric CO2. So on our planet, I'm sure you've heard you hear on the news, atmospheric carbon dioxide is going up in our planet. And we know this by measurements that have been done on Mauna Loa at the Earth Observatory by a gentleman named David Keeling. This is the famous Keeling curve. So as the temperatures are warming and the ice sheets are melting, some of this is in response to changes in the Earth's greenhouse gases. Carbon dioxide on our planet is going up. So our planet, in terms of when we think about what the planet's atmosphere is made of, it's mostly made of oxygen and nitrogen. These are two relatively inert gases, and there's a tiny bit of CO2, very small. In fact, there are some commercials that you might have seen recently. I think I saw them the last time a few years ago, talking about how CO2 going up in the atmosphere is a good thing because plants breathe it in. And that's true. But too much CO2 is not a good thing. It's going to warm our planet. This tiny amount of CO2, 0.04%, is incredibly important for life on Earth. So we are thankful for some of this CO2 because without it, it would be very, very cold. But let's look at some of the trends that we're seeing. So this record began in 1958 atop Mauna Loa Earth Observatory by, like I said, a gentleman named Keeling, whose last name was Keeling. The red line or the black line in the center, this is sort of the annual average through the data. And the red line is this sort of seasonal wiggle. This is the vegetation growth. This is what we're seeing here is the Earth sort of breathing CO2. In the Northern Hemisphere is where most of the land is on our planet. And so during the summer, vegetation grows and it takes in CO2 as plants photosynthesize. And in the winter, it exhales the CO2 sort of back out as things respire and stop growing. So we see these seasonal changes, but the overall trend, of course, is we are seeing an increase in CO2 on the order of about 3 to 4 parts per million per year. When the record started, it was at about 318. When I started graduate school, we were at about 380. Today, we are at about 415. And the rate of change is likely increasing. So this change in the slope is changing. And so we are seeing an increase in CO2, and this is going to continue to change the warmth of our planet. The significant number, so I'm going to, on this next slide, a couple of years ago, I think it was 2017, we passed this threshold of 400 ppm. And this isn't a significant threshold. There's no sort of magic number to the Earth's CO2 being above 400. But historically, the last time that CO2 on the planet was this high was millions of years ago during a period of time called the Pliocene, and the Earth was significantly warmer. So when we look at Earth's history outside of the modern instrument record, we know that periods of time where there was more CO2, we had a warmer planet. So in terms of the need for climate proxies, when we look at the instrument record, we don't have a lot of data. The instrument records are brief. We've been taking the temperature of the planet for 140 years. We have sea ice extent data that's, goes back about 40 years, and CO2 measurements began about 60 years ago. It's not a lot of data. And so because of that, we seek to find information in fossils and in parts of the Earth climate that we can extract data from. Proxies are tools that we can use to take the temperature of the planet in the past. And that's what forams are really good for. So this sort of brings me to some, introducing the field of climatology. I do paleo oceanography. That means I study changes in the temperature and things like pH in the ocean. But scientists study the climate of the past. This is the broad field of paleo oceanography using proxies. Proxies are tools that we use to take. We can't travel back in time and take the temperature of the Earth, but we can obtain fossils and infer changes in temperature in the past. And we do this because if we understand natural climate variability, it helps us to predict future climate change under natural change and as we modify our planet by polluting and doing things with greenhouse gases, we're able to understand how under different scenarios the climate might change. So the figure here that I'm showing, it looks pretty complicated, but I really like this figure for various reasons. This is the temperature of the planet over 500 million years. Oops, I'm going ahead. And it looks complicated because at the bottom, the time changes. So I'm not going to spend too much time talking about this. What I really wanted to point out is that we have a significant amount of information about the Earth's climate history from proxies. Things in the blue colors here, these are temperature measurements inferred from tiny bubbles of gas that are trapped in ice cores. The black line that coincides with the blue here and extends back through this record, and the green line here, these are from forams. These are from fossil foraminifera obtained from the sea floor. And so we can look at this and go back in time and understand that the Earth's climate has been different in the past. And if we understand what controlled that variability, we're able to understand what's going to happen in the future. So I'm going to come back to this section of the graph in a few minutes. And so I wanted to just touch before I leave this slide and just sort of highlight that this period of variability here is the sort of pacing of the ice ages. This is, we are in a period of time we are in an interglacial. We have relative climate stability in the modern world. This lasted for about 10,000 years. And then as we go back further in time, this sort of seesaw pattern is the ice ages. And so I'm going to show this again, but I'm going to highlight how forams play a role in this record. Okay, so I'm going to switch gears here and talk about how forams are really useful for reconstructing the Earth's climate in the past. So the previous slide that I showed, the record here, the record here is from forams that lived on the bottom of the ocean. We call them benthic. They live in the benthos, the sediment that's at the bottom of the ocean. I study the forams that live at the surface of the ocean. They are, we call them forams for short, I've said this already, but they are single-celled protists. They occupy sort of the upper couple hundred meters of the sea surface. And what makes them really useful for paleoclimate research is they have they secrete around their cytoplasm, which I'm showing in these pictures, a calcium carbonate shell. This is the mineral calcite. And I'm going to show you in a minute how this ends up playing a role in their utility in paleoclimate. But they're cosmopolitan in the ocean. That means that they're pretty much found everywhere, and they're very common in the fossil record. So the figure that I have on this plot, it looks like temperature in the ocean, but what I'm illustrating is all of the dots in here. These are seafloor sediment samples that a researcher counted the different foram abundances as. So these are the number of species, and they tend to coincide with temperature. So where we have the warmest temperatures in the ocean, we have sort of the greatest number of species. And as temperatures decrease towards the poles, the number of forams go down. Interesting to the region off the coast of Oregon here, there aren't that many forams off the coast of Oregon here. It's not that there aren't many forams, I misspoke. The deepest Pacific Ocean is very corrosive to the mineral calcite. And so there's a significant amount that the shells can dissolve. And so that would skew some of the data. And so they opted to leave this out of their reconstruction. But the point here is just to sort of, drive the sort of concept home that they really are everywhere in the ocean. And what makes them really useful for paleoclimate reconstructions is that the composition I mentioned in the previous slide, they're made out of the mineral calcium carbonate. The ones that I've shown so far, the photographs of the ones that I've shown so far, those are living forams. And what I'm showing on this slide here are fossil specimens that were obtained from sediment cores. This one here was one that I picked from a sediment sample that was about 30,000 years old. So these represent paleo samples. So they're made out of the mineral calcite, but when they grow, they incorporate small amounts of other elements into their shell, and they do so as a function of their growth conditions. And so as an example, I wanted to just illustrate how one of these tools works. The probably most widely used proxy for reconstructing temperature in the past, we actually call this a Paleothermometer, is to measure the magnesium-calcium ratio in planktic foram species. When they grow, the amount of magnesium that's incorporated into their shell varies with the temperature that they calcified at. And so I can obtain samples from the sea floor, and I can measure the amount of magnesium in their shell. And if the magnesium is low, on the order of a 1 to 2, a ratio of 1 to 2-ish, in terms of their magnesium to calcium ratio, that specimen grew at a colder temperature. And if there's more magnesium, it grew at a warmer temperature. And this is how paleothermometry works. I can obtain fossil samples and literally take the temperature of ancient oceans. And I'm not going to talk about these additional proxies here, but we are able to do things like reconstruct the pH of the ocean using different elements. We can look at the salinity in the ocean, even changes in oxygen, and how productive the ocean was in the past, all using these tiny sand grain sized seashells. So I wanted to illustrate how this works. This is the next couple of slides were lent to me by a research scientist at the University of Arizona. This is a schematic of forams living in the ocean, highly schematic. They live at the surface of the ocean, they grow, environmental data is recorded into the chemistry of their shells, and then they die and they sink and they settle on the ocean floor. And they do this over tens of thousands of years, thousands to hundreds of thousands of years, and even millions of years. They settle on the ocean floor, and then in the modern ocean, scientists can go out on a coring ship, like the JOIDES resolution, and they actually deploy a coring mechanism to the bottom of the ocean floor and obtain these long cylinders of ocean sediment that are quite literally miles long. There are core repositories at Oregon State and other locations in the United States, Lamont Doherty, there's one in Texas, where scientists can then obtain some of these seafloor sediment samples and take them back to the lab and obtain forams from them. So the picture that I'm showing here on the bottom, this is a core that we sampled at the core repository here at Oregon State. It's from the Santa Barbara Basin, and you can actually see some layers in it. So we take about 1 centimeter of sample, and within each centimeter, depending on the location where your sample was taken, there can be up to thousands of individual shells, and we can resolve the Earth's climate in the past to monthly resolution, and then in the deepest ocean, where not many forams fall, I should say, maybe we get about 1,000 years of resolution per centimeter. So we can take the samples that we obtain from these types of sediment cores, pick the forams out, measure the magnesium in their shells, and measure the temperature of the ocean when they grew. So this is the figure that I said that I would come back to, and I promised after this is, I think, my last figure that I'm going to show you this data, and then I'm going to show lots of fun pictures. This is a reconstruction of the temperature over the last 400,000 years, measured in foraminifera, in a foram species. So some scientists obtained I should orient you first. Age is on the bottom, but paleo oceanographers like to confuse people by flipping the axis of the time. So when you're looking at the age here, it's actually modern is on the left, when it makes more sense to think about time being in the past. So we're actually looking at the modern ocean up in this corner. This is where we are today. We're in a relative warm period. And going back through time, the last glacial maximum, and then going back up through time here. So we're actually seeing the seesaw pattern of the ages recorded in the temperature of forams that were obtained in the central Pacific Ocean, or in the tropical Pacific. They measured the magnesium calcium ratios, and from that inferred the temperature that they grew at. What was really outstanding about this study is that they were able to show that the pacing of the changes in temperature that's recorded in their shells coincides with temperature reconstructed in ice cores in Antarctica. So we're seeing global temperature change in 2 locations by using 2 different proxies. Pretty outstanding. So I want to transition from looking at data to looking at how we do this. How does my research play a role? So I started out studying paleo oceanography, and my students in my lab still do this. We obtain sediments from the seafloor, and we do paleoclimate reconstructions. But we also study recently live forams, or ones that we capture in the ocean and we allow them to live out the rest of their life cycle in the lab. And we do this to really fine tune how we can use their shells in the fossil record. We call this Paleoproxy development. And I'm doing this with species that are sort of commonly used in the fossil record, but that we are not commonly grown in culture. So I think there's a lot that's still left to learn about them. And of course, these are pictures that I took of ones that I grew, and I'm going to talk to you now about how we do this. Why do we want to grow forams? So we can control their growth conditions. We can do this, we can grow them under really tightly controlled conditions, and then measure the chemistry of their shells to see how growth conditions change the composition. And then we can take that information and apply it to fossil specimens. We also do this to understand how do they grow? How do they biomineralize? This gives us information about how resilient they are, for example, to climate change. As the pH, or as the atmospheric CO2 is going up, the pH of the ocean in response is going to go down. And this can make it harder for organisms that make their shells out of calcium carbonate to grow. So we do this so we can understand how sort of modern climate change might affect their ecology. So how do we do this? We capture them by plankton nets. So I'm going to show this in a minute, and by diving, and then we grow them in the lab. So I like to joke around when someone says, why do you grow forams? And I say, well, because I get to go to these great places, which is a little bit of a joke, but it's really true. I get to go to some really great places to do field work. So during my postdoc, I went to Catalina Island and did most of my initial research at the Wrigley Marine Science Center, which is sort of tucked into this cove right here. And we hop on a little dive boat and go past this rock. And within 15, 20 minutes, we're in deep water collecting forams. I did this over about 5 field seasons on Catalina Island. In 2019, I had the opportunity to travel to a tiny island off the coast of Taiwan called Green Island. Spectacularly beautiful, probably one of the prettiest places I've ever been to. It is an 8 square mile island, very, very small and very hard to get to. It literally took planes, trains, taxis, a ferry. And then once we got to the island, we get around the island by driving scooters. It was probably one of the funnest places I've ever done field work. And then since I've arrived in Oregon, we've had the opportunity to go out on some ships off the coast of Oregon to collect forams that live in cold water. So this is a region that is really understudied in terms of collecting living specimens. And you look at Green Island and you look at coastal Oregon and you think, well, sometimes it's pretty dreary here in Oregon, but I've got to tell you, the sunsets that we see and sunrises off the coast of Oregon on a good cruise are pretty outstanding. This is a sunrise one morning when we were on our last cruise. And then these were 2 of the sunsets that we got to see. This was just last September. We got to go on a cruise. Even during the COVID lockdown, we all had to mask up and quarantine for a week. So we do still see some pretty spectacular views in the Pacific Northwest. So how do we catch the forams? So off the tall ships, or the big ships off of the coast of Oregon, when we go on the Ballon Shimada with my colleagues, or the Oceanus, one of the ships that's run by Oregon State, we collect them by using plankton nets. So this is a photograph of my graduate student, Kelsey, and myself deploying our big foram net. This is our biggest foram net. It's 1 meter. So it's quite large, and it's deployed off of a crane that then goes down and comes back up, and we pick the forams out that way. When we go into the field in small field work groups, like on Catalina Island down here and Green Island, we're actually on dive boats. It's a much, much smaller operation, but for an extended period of time, we are usually in the field for, a month to 6 weeks or so. And we are deploying things like plankton nets, and we also deploy something called the CTD, which measures the temperature and the salinity of the seawater. And we do this all by hand. And yes, we do deploy our 1 meter net with a huge opening behind a boat, and we all pull it in. And our crew in Taiwan was nearly all women. And yes, we all come back from the field with big arm muscles. I'm not kidding. It's not an easy task. When we were in Green Island, we had an epic field day where we put the net and the CTD in the water enough times that the amount of line that we put out and pulled back was about 1 mile in length. So it was an amazing day. And then the last way that we collect forams is by picking them out of the seawater by diving. So I'm usually topside on these boats operating the nets, and we've got a crew of amazing divers that go through some pretty rigorous training to be able to do what's called blue water diving. This is one of my collaborators, Oscar Branson, hanging. He's tethered to our boat in Taiwan, and in his hand, it's a little bit hard to see, but that's a little glass jar that we literally, they can view the forams and capture them in the jars above them. And then the images on the bottom here are from Berbel Honish, who also does culturing. These are Puerto Rico. And so they're all in short wetsuits because it's warm there. But I love this picture because it gives a really nice idea or a sense of what the divers are doing. They go down in groups of 3 or 4. Here's the ship, and or the dive boat, and they're tethered to it, and that's the goodie bag that they collect forams into. And then I think this little pole right here is a shark stick. So no, no one's been approached by a shark before. I think it happened once in Puerto Rico years ago to my postdoc advisor, but it's pretty rare. So after we collect the forams, we bring them back to the lab, and they all go into their own glass jar for growing. So each foram has to get taken care of the entire duration of its life cycle. This sounds a little bit ridiculous, but we have to control its temperature, the feeding. We want to know how long it lived in culture. We want to know what size it started at versus what it ended at. So we have to track every single specimen. And this is a picture that I'm showing. These are the specimens that we were growing in Taiwan. Each jar has one foram in it. And then the dots at the top, this is pretty fun. This is how we keep track of whose experiment we're working on, because we all sort of take turns. And then this video here, I'm going to pause this and start over. This is how to feed a foram. So we have to feed them. We actually hatch brine shrimp, so monkey shrimp, sea monkeys. We buy them from Amazon.com for $5. We hatch them for 1 day, and then we take the newly hatched little Artemia, and we pipette them onto the foram. Now, in this case, the foram is very large. This is the largest species that we work with. And I took a video of this, very, very high tech. This is a video that I took with my iPhone in Taiwan a few years ago. So this is the living foram at the bottom. Around it is its spines. And I have a little glass pipette that I'm sending the live Artemia down. And once it even gets just close enough to the foram, it sticks to its sticky rhizopodia. And then within a couple of minutes, the little Artemia is actually pulled in up against the shell, and then it gets dissolved and the foram eats. I have a video of this. If you're on Twitter, I have a video of this pinned to my account, and you can watch sort of a high resolution video of one that I accidentally fed to, and you can sort of watch the Artemia get pulled in. So we put them in culture, we feed them, and we do this until they go through something called gametogenesis. And this is basically foram reproduction. This is a different species this time, and I'm going to hit play here and show you what Gamet release looks like. So this is a foram that's going through reproduction, and it's releasing gametes. There's almost nothing inside that shell anymore except gametes. And this is what I would consider fairly explosive gametogenic response. Sometimes they don't look like this. It looks sort of like this sad blob of gametes that sort of is pushed out of the shell. This was one of the first times that we had ever seen this happen in the lab. And if I were to play the sound, we were so excited that I think it's a fun video to listen to if you know what's going on, but this is one of the first times that we'd ever seen this before. We have not been able to get multiple species to go gametogenic together and make foram babies yet in the lab. If we could do that, it would change our ability to do the type of research that we do. But so far, something about their life cycle is missing. We can't get them to produce gametes and make foram babies. So this is why we have to painstakingly take care of each and every one of them. Okay, so for the last couple of slides of the talk, what I wanted to do is give you a sense of how an additional sort of utility of forams. We've been studying forams off the coast of Oregon. For the last couple of years, I've been going out and collecting samples, but one of my graduate students, Kelsey Lane, had this really wonderful opportunity to go through some plankton tow samples that have been stored at the Hatfield Marine Science Center and look at an archive of samples that had been collected over the last decade. And so I want to show you how we're using this archive to understand how rapid foram assemblages can change in the modern ocean. So for some context, I'll walk you through sort of where this study's happening. We're doing the research in samples that were taken as part of, or that were obtained through a study that coincides with the Newport hydrographic line. So this is essentially this region off the coast of Newport that has been studied pretty extensively by lots of scientists. The one here, this is a set of plankton tow samples that were collected by a gentleman named Bill Peterson and his research group, and that work has been carried forward by by a collaborator and Kelsey's committee member, Jennifer Fisher. Copepods have been studied in this region. Copepods are very important. This is what large fish and whales eat off the coast. So they had been studying or collecting these samples to look at copepods, but they have forams in them. So this was sort of this opportunity to go through these archived samples and generate data to look at what's been happening off the coast of Oregon over the last 10 years or so. So the samples had actually been collected. This sort of archive of sampling began in 1960, but Kelsey's work so far has focused on fall samples obtained over the last decade, so since 2010. And what she's doing here is essentially an assemblage study. So she's going through these samples and picking out all of the forams and identifying what the species are and then counting their abundances. And this gives us an idea of what assemblage is present at this given moment in time that the plankton net was collected. So what was really interesting about this opportunity is that in this region over the last 10 years, two sort of significant marine warming events happened. So one of them was the marine heat wave that occurred in 2014, or it began in 2014. It lasted for about 3 years. A marine heat wave is essentially a warming event that happens in the ocean where sea surface temperatures warm significantly above average. So in this case, temperatures were 3 to 4 degrees warmer than normal for an extended period of time. The first marine heat wave event began in September of 2014, and it was colloquially called the blob. So for those of you who are from the Pacific Northwest, you've probably heard this before. It's been in the news. The first marine heat wave lasted for 3 years. And then there was a shorter duration marine heat wave that sort of returned in 2019. And it stuck around for almost a full year. It was still moderately warm in 2020 when we did our last collection. And then it dissipated and it is essentially offshore now. So this gives you sort of a snapshot of what the temperature looks like off the coast. So to orient you to the figures, these are sea surface temperature anomaly maps. So this is relative to sort of an average temperature. The reds are warm, the blues are cold, and then the whites are right in between. And this is the Newport, the region that we're focusing on is right where this Newport hydrographic line is. So September of 2014 and again in 2015 look really similar. It was significant warming. Key difference is that in September, the warm water was very restricted to sort of the top of the ocean, whereas in the previous warming event, it lasted for a few years. And then off the coast now, there is still some warm water. The black line sort of shows where the warm water is. And this region here in the dotted blue line is sort of the United States coastal waters. And I've put a link here if you want to learn more. There is actually a blob tracker. So they're keeping track of what's happening because during these events, ocean conditions change enough that they're changing the food web. So this is something that's of interest to a variety of scientists. Okay, so I would like to sort of walk you through the results. It's a little bit of a complicated figure. So I'm going to sort of explain what this is for the Foram people who are here. I know there are a few of you. The species names are in the legend here, but what really matters is the colors. So in 2010 and 2012, this is sort of typical Oregon. We see a significant number of species that are typically found in cooler waters, and the abundances were actually quite low. The plot here shows relative abundances, so it's a percent. And then each one of these bars represents a plankton net going from the nearshore region offshore. So if I go back to this plot here to orient you, the nearshore region is 25 nautical miles offshore, and the study goes all the way out to 200 nautical miles offshore. So in 2010 and 2012 is pretty typical Oregon. There have been folks that have studied this region, decades ago. These are species that are common to the Oregon coast. But in 2014, with the onset of the marine heat wave, the species changed. So in 2014, we have by far warmer species. The first thing you notice here is there's no blue. Sorry, my mouse has stopped working. There's almost no blue. So we see a transition from this cold year in 2012, fast forward to 2020, and we see the colder water species are gone, and we're starting to see species that are more common sort of off the coast of, Southern California, Catalina Island. And it's not that these species aren't ever found off the coast of Oregon, but they're typically very, very offshore. So we wouldn't see them all the way onto the sort of coastline of Oregon at this location. Transition into 2015, this was sort of peak warming in the marine heat wave. And at this point, we start to see subtropical species that are not common to this region. So it's not that they're not found here ever, but they're typically not found across the entire NH line, across the entire sort of sampling area. The reds here are species that are quite common in the tropics. So we're starting to see some warmer species. This is 2015. 2016, we see some warmer species again. This is when the marine heat wave started to dissipate. And then as we transition into 2017, we're out of the marine heat wave is dissipated, and we are back into cold water species. So now I'm going to show 2018 and 19 together. And 2018 was an interesting year because it was sort of the onset of the warming coming back. And what we see is this transition off the coast from the nearshore environment where we're seeing colder water species to the offshore environment where we see warm water species. And in 2019, we're back to this warm event where we're seeing species that are more common off the coast of Southern California where I've done my culturing. So again, I'm not saying that these species aren't found here, but they're typically far, far offshore in sort of the gyre waters that are, thousands of miles off the coast. So what we're seeing here is rapid changes in the species that are present off the coast of Oregon in response to warming. And what we can do with this data, interestingly enough, is now that we know that we've seen these species here, we are going to look for marine heat waves and sediment cores that we've obtained off the coast of Oregon and see if we can't do a climate reconstruction to see if we find these species in the sediment cores, we can tell how frequently they've happened in the past. And if we don't find them at all, that would suggest that what we're seeing today is pretty unique to the modern setting. And I will tell you that we don't have the data on here, but 2020, we were finding tropical species off the coast of Northern California, just south of the Hecate Bank. So if it's warm enough, these species are there. And so I'm going to end my sort of science talk there. On this next slide, I would like to first thank the 500 women scientists for giving me this opportunity to come talk. In COVID times, this is a pretty interesting way to talk science. On this plot here, on this figure here, I'm going to just let this play. This is the global temperature change over the last 170 years or so. And then I've given some access to where you can go learn more about what's really warming the world. Is it humans? Is it natural? Yes, it's humans. Yes, it's us over natural climate variability. If you don't know who Katharine Hayhoe is, I'm sorry, my slide has a mind of its own. If you don't know who Katharine Hayhoe is, you should. She is a climate science communicating champion. She's outstanding and has a really unique way of communicating science in a manner that everyone can understand. And then I've given some resources for outreach that we've been working on in my lab. And I would like to, of course, thank the 500 women scientists again and my funding sources. And I've got contact information on the bottom, and I'm going to leave that there. Thank you.
Yay. Thank you, Jen. That was great. I love Tap Talks. I learn so much every time from you and also from Susan at the very beginning with her trivia. But
yes, that's
right. I, will always be swimming with Forams forevermore in search.
That's right. If you swim in the ocean, you have probably swam with Forams. If you walked off the coast of Oregon in the mudflats, you've stepped on them. They're there.
So as a group, if you can, just cheer, clap, in your own living rooms. We can't hear you. It's not.
I can't hear you or see you, but I hope you learned something today.
When we usually live in non-COVID times, we're at the tap room at block 15 and we pack out the place. And so then everybody's together. So this is very different. And I'm so grateful that so many of you showed up and were here and participated. This has been going so great and just warms the cockles of my heart because I'm British like that. we're going to open up the floor to Q&A, which Ashley Antonio will actually facilitate because she's much better at that than me, with lots more experience. Before that though, before people disappear, just a heads up, our next tap talk is on March 2nd, which is also Tuesday. It's always first Tuesday. Our talk will be about coastal pollution. It's not just the microplastics, it really isn't. And that will be given by Suzanne Brander, Sarah Hutton, I'm going to make a mess of this name, Samreen Siddiqui. And so that would be great. So this is a different change up. This is giving graduate students an opportunity as well to actually present and give tap talks, because we're all about making science open, inclusive and accessible, and it's a great opportunity. So please tune in next month to listen to that talk, and you'll learn lots about more than microplastics, I'm sure. Also know that this talk was recorded. We now have a 500 Women Scientists Corvallis YouTube channel. Thank Yvonne Richer for that. Just search YouTube, you'll find it. We'll upload in the next couple of days, and you can actually start catching other tap talks as we put them up. We're learning how to do this as we go. Anyway, again, another big thanks to Jen for being such a brilliant, fabulous, wonderful presenter.
Thank you.
And I will open the floor to
questions. Ask me anything.
Yeah, so it's kind of large groups. If you have questions, putting them in the chat might be the best way to go. So I haven't had any questions in the chat yet, but you can just type those in the chat and I'll help facilitate those questions to Jen. And while folks are typing, if you didn't notice, Natalie put a knitting pattern as a forum in the chat. If any of you are knitters want to knit your own.
Fabulous. That's fantastic.
So that's in there. And so that's pretty fun. I guess we're waiting for folks to put questions in the chat. I was wondering, Jen, if maybe you could, I don't know, I'm curious, like, what is your most favorite part about this research? Like, what's the most exciting bit that you get to do, or what's most exciting to you?
Ooh, that's a good question. So I think the most exciting part is especially when we culture forams, or even just measuring the fossils. We don't know whether or not an experiment was successful until we get back to the lab. So probably one of the most exciting pieces of, or like exciting experiences that I had was conducting this sort of really unique experiment in the field. And then you don't know it's going to work until you get back. And months later, you clean them and you analyze them. And I literally shouted in the lab because I could tell as the data was coming off the instrument that the experiment worked. It was really exciting. So I think that's really fun. And then seeing things happen for the first time that we've never seen before. And this happens all the time. We talk about it. We've had videographers in the field with us, and they will tell you that they get excited when we're like, oh my gosh, someone come and look at this. And because we see things really that are unique for the first time. And that gives us insight to their behavior. And yeah, there's a lot of really fun stuff when we're looking at the live ones.
Great. We have some questions coming in the chat. So Tom and Lori asked, how and when were forams discovered?
Ooh, so forams have been known for a very, very long time. So some of the historical data actually comes from Darwin, right? And like the early research cruises. There are some phenomenal artwork pieces that have been done by early researchers who have had these are folks who are talented enough to draw them with the most intricate detail. And you can buy the prints and put them on your wall. It's amazing. But forams we've known about them for a very long time. I think people looking at seawater under a microscope.
Great. Maura asked that, so they say, I'm guessing multiple projects or researchers use these cores. So how do you all decide how many bytes can be taken out of them, seeing as they're essentially a finite resource?
They are a finite resource, and sometimes that's a little bit of a bummer. So with ocean cruises, the folks that go out on the big ships, like the JOIDES Resolution, the coring ships, they get first dibs. So they actually have what they call a coring party. They all get together and they put little flags on the cores, and they obtain all the samples together. And then the rest of the core sort of goes under, it's not allowed to be used for a certain period of time. Usually that's about 2 years. And then the cores go into a repository, which those repositories, there are several in the United States. There are some that are overseas. And then you can look for the core online and put a request in. We take the core and cut it in half, and one half becomes the working half that people take samples out of, and the other half becomes the archive half. And this first working half is consumed first. And then over time, they go into the archive half. And after a little while, there's nothing left. And that does happen. So sometimes you will put a request in. This just happened to my graduate student. She put a request in and they said, we can't give you any sample because there's nothing left. If this happens in a very important region, then scientists will get together and try to get another coring rig to go out. But yeah, there are only a finite number of samples that can be taken. We do share, so if somebody says, you take this piece and you take this piece, even out of the sediment samples once we process them.
Great. Keith asked, how does ocean acidity complicate paleoclimate predictions using Forams?
Yes, that's a really good question. So ocean acidity in events that have happened in the past, where the ocean pH has changed significantly, forams actually completely dissolve. So there are locations in the ocean that it actually looks like we call it a burn down. You can actually, the PETM with some of them, I'll put this in the chat. If you just Google the PETM and sediment cores, you'll see a picture of this. This is a gentleman named Jim Zakos who obtained the cores, and you can actually see a transition from sort of white sediments to clay because the forams are gone. When the ocean warms rapidly, or kind of what's happening today, there can be a foram extinction event where you drive the foram numbers down to very, very small numbers, and then there's sort of a evolutionary event where others take their place in the recovery period. In the modern ocean, what we're predicting will happen is that forams will eventually have a harder time calcifying and they become thinner. So ocean acidity makes them thinner and it makes them more prone to dissolving on the seafloor.
Great. Lauren asked, you mentioned you take a 1 centimeter section of the cores. How do you decide which bite or bites to take? Is it just the most recent, the one closest to the top, or what's that decision-making process?
Oh, that's a good question too. So the core top is probably the most interesting because it has the forams that were most recently alive. So we can get as close to modern climate in those samples. And then in terms of the sampling frequency, it depends on a bunch of different factors. Off the coast of Oregon, for example, we just took samples out of a core. We literally took every single slice because the sedimentation rate is really low here. I want every bit of data. But in a core from the ocean, if I've got miles of sediment to go through, I'm going to maybe pick every 20 centimeters. And then I'm going to radiocarbon date those or use some other mechanism for getting an age correlation. But yeah, so it kind of depends on the questions that we're after. But the core top is probably the most significant for almost any researcher, unless you're skipping the modern and doing a time slice in the past.
Great. Mary asked, how did you get interested in paleoceanography?
That's also a really good question. So when I went, so in terms of sort of like my education history, I have a degree in geology, and then I went and I worked for 5 years. And I was really, really longing to get back into science. And I was working in IT. I completely left science. And then the .com field collapsed and I went back to school. And I met who would turn into be my advisor, Dr. Pam Martin, at the University of Chicago. And she said, hey, I have this project. I did a small project on the biogeography of ahermatypic corals for my master's, totally unrelated to what I do. And then she said, hey, I've got this really interesting idea, you looking at forams. And I was so green. I didn't understand really what made their shells look the way that they did. And sort of getting to the punchline, a couple of years into my degree, I learned that the composition of their shells is actually really complicated. It's highly variable in single shells. And that just fascinated me. This is what got me into culturing them. I wanted to know why. and the history of the Earth's climate past. I mean, that's just really fun. It's like little time machines.
Great. We're coming up on 8, but I think we have time for a couple more questions. So Jim Mitchell is asking, and I think this is in reference to the Oregon, coastal Oregon study. Were you surprised at how quickly the populations changed?
Yes, absolutely. So I'm not surprised. Forams actually have a pretty tight temperature threshold that they can survive in. Each species has sort of this range that is their sort of happy spot. So I wasn't surprised when we got into warm water that the cold water species were gone. I was more surprised at how fast we saw the tropical species in this region. And so we've been working with physical oceanographers and modelers to try to figure out where this water, the entrainment of water, might have come from. Yes, we're very surprised. And I will tell you, in one of the research cruises that we were on, we went from being in the warm water in 2019, and we were sampling off the entire west coast of the Oregon from Washington to California. And we transitioned out of the warm water, and the next plankton tow sample that we had in the cold water was completely different. So it really is just being controlled by their temperature. And so, yeah, I was surprised at how quickly they changed. Absolutely.
Great. We only have 3 questions left, so I'll try and get through those if that sounds good. So the next question is from Noah, and that's asking if there was any form of stimulation that caused the release of gametes in the video that you showed.
That's a really good question. I don't really have the answer to that. That season, that field season in particular, almost all of them behaved that way when they went through gametogenesis. So it was a really warm year. So it might have been the energetics of just them, the higher metabolism. Yeah, so I actually don't know. And we still do see that. So we've seen it in Taiwan. It might have just been luck of us looking at them at the right time, and now we know better. Yeah, so no, I don't think there was any stress that caused it. I think it's just sometimes they have an explosive gamete release and sometimes they don't.
Great. So Dave asked, what other physical and chemical measurements can be determined by proxy from Forams?
There is so much that we can learn from Forams. We can measure boron and infer changes in pH. We can look at sodium and infer changes in salinity. We can look at oxygen content in the ocean in the past, productivity, nutrient utilization. It's pretty wild, and some of these things are changing. So as we get better instruments, we can ask different questions, and so the field sort of continues to evolve. But yeah, we can get a lot of information from them.
Great. Next question. David says, the new administration is putting a lot of work into combating climate change. Is there any action that's going to best impact ocean warming?
Stop burning fossil fuels.
Great.
I mean, if we stopped emitting fossil fuels tomorrow and went to 0 emissions, the planet is still going to continue to warm because it hasn't caught up yet. So even if we stop emitting CO2 tomorrow, we're in it for warming for a little while. And the best way to fix what's happening is to stop fossil fuels. We have to go to clean energy.
Great. And Brian Lee snuck a question in there, so we'll make this the last one. And of course, there's lots of words that this recreation ecologist is probably going to butcher when she pronounces them. But do you use image recognition micrography to identify and count the foraminifera in samples?
Yep, the forum samples.
Yep.
Yeah, this is a new growing area of folks who are using basically machine learning and AI. And I was actually part of a study. The lead author on it is trying to create something called the Forabot, which is like a Foram robot identifier, and the machine does a better job than humans. So we can use machine learning to do Foram counting and identification. Absolutely.
Great. Well, that was our last question, and we're right at 8 o'clock. So thank you everybody for the great questions and the engagement, and thank you again, Jenn, for the wonderful presentation.
Thank you, everybody.
I learned a lot. And again, next talk in a month. So we'll see you all in March, hopefully. Have a great evening.
Bye, everybody. Thanks for coming.